Now we have the monocot leaf, and when we study this type of leaf, we are going to keep in mind what we have studied in dicot leaf, and the points of differences are going to play a key role in this lesson. Now, because this is a leaf, the anatomical features would be almost similar. The points of differences would be important to note down and understand. So let us see what the anatomical differences are present in a monocot leaf, how it is different from a dicot leaf, and a few terms that we'll come across. In the previous lesson, we had talked about the upper surface and the lower surface of the leaf, that would be comprising of single layered epidermises, both at the upper and the lower side. A few new terms that you would get introduced to, which are applicable for both the cases because they are universal terms. Firstly, the upper surface of the leaf is known as adaxial surface. This is a term you need to know it. In case you know it, that is quite good at your part. You're a well-read student. In case you don't know it, the purpose of these classes is making you a well-read student by the end of the lesson, so you can remember it. We have the adaxial and abaxial surfaces, or otherwise known as the upper epidermis and the lower epidermis. Like in the previous case of a dicot leaf, we had the mesophyll that lied between the both epidermises, and the mesophyll over here, unlike dicot, is some sort of is showing some sort of uniformity, as you can see. So the mesophyll is not differentiated into palisade or spongy parenchyma. Instead, there is a uniform mesophyll that is present between the two surfaces. I would label it on the other side because we have to take into consideration the vascular bundles as well. So it would be easier at that side. So what you see over here in the case of mesophyll is that the intercellular spaces are few. It is not having air spaces as in the case of spongy parenchyma. It is almost uniform. Alright. So the mesophyll is almost uniform. There is no distinction of palisade or spongy parenchyma. And in between the mesophyll, there is a parallel venation. We come to know about parallel venation from the morphological aspect of the leaves. Like in the case of dicot leaf, you have such sort of venation. You remember your drawings from the lower classes. This is what the reticulate venation means, that veins are forming a network. So when we represent this part, we have a midrib that passes through. In the case of dicot leaf, this is a grass supposedly. Take for consideration banana leaf, or you can say the example of a sugarcane leaf. So what you see, instead of that sort of venation that we have in dicot leaf, there are parallel veins which are passing. So you will find vascular bundle over here. The next one could be located in the next part because this is the vertical section. So you would find many vascular bundles present. Okay, now coming to the vascular bundles, what we have next to understand is that the vascular bundles would be conjoint, collateral and endarc. I'm not going to repeat what it means because we are quite aware by now what these three terms mean. As you can see, this part is xylem. The xylem lies towards the adaxial surface, that is the upper surface, and the phloem lies towards the abaxial surface. So this is one point. What you see over here is that the vascular bundles at the upper surface and the lower surface, they have certain sheets of sclerenchymatous cells. This you have to remember, that sclerenchyma would be present on the upper and lower, just below, beneath the adaxial and abaxial surface. That is, the presence of sclerenchyma is seen, and the vascular bundles, they are like sandwiched between the sclerenchymatous cells. Alright. Another point that you have to remember is that it could be surrounded by bundle sheath cells at times. And one thing that you have to remember, that the grasses at times have bulliform cells. This is to be remembered. Bulliform cells are present in the grasses towards the lower and upper and lower surface beneath the epidermis, and these bulliform cells, they contain water inside them. Now these bulliform cells perform the function of giving the shape to the leaf. Alright. Now this providing shape is taken care by the water content that is present inside these bulliform cells. So they will be parenchymatous only, because parenchyma is responsible for storing. Apart from the nature of bulliform cells, what you can see over here is that the parenchyma, that is mesophyll, they are having oval or circular cells with least intercellular spaces. They are rich in chloroplast, unlike the epidermal layer, which is not having chloroplast and it is devoid of chloroplast. It is colorless layer. There is presence of cuticle, both above and lower epidermises are having cuticle on them. Now there is one point of distinction, that in the case of dicot leaf, what you observed was that the upper epidermis was thicker, was lined by a thicker cuticle, while the lower epidermis was not having that much thick cuticle. In the case of a monocot leaf, we have the upper and lower epidermis is lined by a cuticle which is having equal thickness both at the upper side and the lower side. So the cuticle will not be different in its thickness. So one point of difference is this. The first point that we talked about was that there is no distinction in the mesophyll in the form of palisade or spongy. The second is that the thick cuticle distinction, that is thick, would be present above, and the lower will have thinner cuticle, that is also not present. And thirdly, there is one major point of difference is that we have in the case of monocot and dicot leaf, that happens to be, this is called isobilateral leaf. Now why it is called isobilateral leaf? Over here we are taking the example of any grass leaf. Isobilateral leaf, the arrangement of stomata, or you can say the presence of stomatal pores, is uniform both on the upper and the lower surface. Compare it with your knowledge of dicot leaf, where we had discussed that the lower surface, that is lower epidermis, or to be specific in our new term that we have learned, that is the abaxial surface has larger number of stomata. In case there is a presence of stomata on the adaxial surface, they would be very few to minimal. In the case of monocot leaves, you are going to find the stomatal pores present equally in their distribution at both abaxial and adaxial surfaces. This point you have to remember, that the stomatal pores would be uniform in their distribution in both the epidermises, and the free air space would be minimum. They would be very much less as compared to that in the case of dicot leaf. So we find guard cells present in the upper surface and the lower surface, and stomatal opening is present, which has air spaces from where the cells, or the, you can say, the leaf is going to get rid of the gases, or it's going to take up the gases. So these stomatal pores and the guard cells which surround them are responsible for the gaseous exchange, and they are equally distributed. So the gaseous exchange takes place from there. This is the only cell, the stomatal cells, or otherwise known as guard cells, are the only cells in the epidermis which have what? They have chloroplast in them. Now this chloroplast serves the purpose for performing photosynthesis, which is responsible for their opening and closing. So this is all that you have to know about an isobilateral leaf, that is a monocot leaf. Any grass that we take will have this inner composition. This is the vertical section where you have upper epidermis, lower epidermis. A patch of sclerenchyma surrounds the vascular bundles. The vascular bundles are conjoined, collateral and endarch. What you see in the red color is the xylem, and the blue representation shows the phloem. We have the mesophyll cells which are rich in chloroplast. There are little bit of air spaces that are present. The lower and upper epidermises have equal distribution of stomata. There is presence of bulliform cells which by their water content regulation are responsible for giving shape to the leaf, and the new terms, that is adaxial and abaxial, that you came to know. So this is all about the monocot leaf. Now again I'm repeating, quite repetitive I have gone in this thing, that whenever you are doing this diagrammatic representation, do use colors for your better understanding, and from the understanding point of view, do keep in mind the points of differences.